Why, hello there, gangster. I didn't mean that in a mean way. I meant that in a cool way. Like, I think the cool kids say that. They're like, hey, gangster, what's up? I'm just trying to fit in. So, since the last time I posted a video, I got a haircut. Actually, I got them all cut. So, in regards to my project, I'm at a stopping point. I have my plasmids, and while I could probably make some more for the next part of the experiment, that's done. The next part of the experiment is to take the plasmid that I've been working on and put it into diatoms. This is a process called transformation, which I did with bacteria in order to grow the plasmids in the first place. But this time we're actually trying to get the diatoms to use the plasmid we're working on. And we'll be able to tell if the plasmid is being used by a diatom because of what's called a reporter gene. A reporter gene is a protein that is easily measured in an experiment. In our case, we use green fluorescent protein. Green fluorescent protein, as it sounds, glows green. So, if the diatom is using the plasmid that we gave them, it'll glow green. I want to say grow green. But now I'm preparing for Fall Fest. Fall Fest, which is in early November, is a science fair. It's a science fair for all the students who did research over the summer. So basically, I'll be standing in front of the poster presenting to anybody who stops by. Now, that might be a biology professor or a first year who's studying psychology. So, as you can imagine, the range of people coming by will be very, very different. I'll have to make my presentation fairly general and be able to explain things at a very intricate level and a very, very broad level. But that's really good practice for me, so I can understand all the intricacies of my project and be able to explain it all the way down. It increases my learning. So, to prepare for Fall Fest, I'm going to compose all my thoughts in more or less a paper. So that means writing up my materials and methods, explaining all my experiments and how I got to the point, and how I know what my plasmid actually looks like, and those sort of things. And while it can be kind of a pain to do all this writing, like I said, it's really good to come back and step back from the actual lab work and write down your thoughts and figure out everything and get on the same page. So that's where I am with my research right now. I'm kind of taking a break and working on writing for Fall Fest. Other than that, the only other thing I wanted to mention, which is kind of cool, I'm working on a blogspot. This blogspot will be an area where I post a lot of things. These things will range from videos. There you can see my Twitter feed, and you'll realize that I do spend time outside the lab on occasion. I'm also working on a photo bucket, which I'll be linking to my blogspot, so you can see pictures from campus, pictures from field trip kind of things, and other student life. And there's some other links on there too, like a friend of mine is working on a blog for Clark also, so that'll be there too. So this will be a whole bunch of fun stuff, and I'm looking forward to having one page where you can see all these sorts of things. So I'll go ahead, like usual, and I'll post a link up here. Oh, there it is. It was stuck, sorry. So the link's up there, and that's that. Thanks for watching, and Snarf with the Garthok.